Breaking the wall of slow and inefficient drug discovery. How new drug candidates can be identified faster through automated molecular design. Gisbert Schneider, ETH Zurich. On November the 9th, 1989, I was on top of the wall, right in front of the Brandenburg Gate for most of the night, anxious and excited at first, but then over the moon.
Hello, Berlin. I always wanted to do that. Dear all, our future is green, clean, and healthy. There is no alternative. Our future generations urgently need new medicines. However, their discovery and development is a time-consuming, very costly process. Only one in millions of molecules ever makes it to the market. Finding the answer to the question which molecule we should synthesize is like searching for and finding the perfect grain of sand on a beach. And it is this, the extremely difficult process of finding new drug candidates, that is the focus of my breakthrough. Now, why is it so difficult to find new drugs? There are many more drug-like molecules which we could synthesize than atoms on planet Earth. We are confronted with essentially an infinite number of choices. And add to that the fact that our knowledge of the molecular underlying causes of human disease are limited, they are incomplete. So, our challenge is to pick the best molecules and eliminate those with undesired side effects or low performance. The human mind struggles with this task. There are simply too many choices and constraints to consider. Now let me illustrate this dilemma. Imagine a world of doors. Imagine you are confronted with a task to pick the one door that will guide you on a pathway to the next billion-dollar molecule, a new medicine. Which one is it? It's fundamentally impossible to test all the options, all the choices. Contemporary approaches nonetheless aim to synthesize and test as many molecules as possible. Essentially, a targeted hit and miss strategy. This works, although it comes at a very high cost to success ratio. Our breakthrough offers an optimal solution by inverting the perspective from screening for new molecules to designing new molecules, from screening to design. In our very effective method, decision-making is partially transferred from the human science manager to an artificial intelligence, a virtual chemist, which is able to generate new drug candidates on demand in an autonomous fashion. This artificial intelligence designs new molecules from scratch and eliminates the need for screening huge numbers of chemical substances in costly experiments. Now, in our world of doors, this means that the algorithm points us to a promising path by generating a new door. By designing new molecules, the chances of finding useful, successful drug candidates are massively improved. Studies from my lab and industry projects suggest up to 10,000-fold increase in efficiency. Better drugs faster with machine intelligence. Now, I started working on this idea approximately 30 years ago, actually here in Berlin. But it took a quarter of a century for our original idea to mature and eventually gain impact. I'm incredibly grateful to my phenomenal research teams and partners who held the line over all these years. The first one through the wall always gets bloody, always. This quote from Aaron Sorkin's movie Moneyball, possibly my all-time favorite, adequately describes the situation we were facing until recently. The main challenge to overcome was a managerial task, namely to cultivate a new mindset to embrace change. It was unimaginable that a computer model could actually generate new drug candidates, possibly even better than a human chemist. Now, how does it work? How does this breakthrough technology work, this new concept? The underlying technology works similarly to automatic text expansion on your smartphone. Our algorithm designs new molecules like typing new words. It does not extend new words, but it generates new molecules. The algorithm is based on the collective knowledge of tens of thousands of chemists. It learns from textbooks and emulates the creativity of human scientists. But it takes it one step further, namely from syntax to semantics. It does not consider only the chemical structure of a molecule, but also the molecule's potential biological activity. This is achieved by regarding a molecule not as an isolated entity or word, but by considering the relationship to the world of known drugs. Let me speed things up. I'm convinced that this smart combination of mind and machine will deliver the next breakthroughs in drug discovery. We've only just scratched the surface. Thank you very much.